right. In the interest of making sure we leave plenty of time, why don't we go ahead and get started? Welcome everybody to the GloBIAS seminar series. We're delighted today to have, as part of our infrastructure series, the scientific added value of a core facility dedicated to bioimage analysis services. I will be chairing for you today. I'm Beth Simony. I'm the Associate Director for Bioimage Analysis at the Broad. And I'm delighted to introduce you to Jean-Yves Tinevez from the Institut Pasteur. A couple quick notes about GloBIAS before we get underway. If this is your first time joining us, welcome. GloBIAS is a registered nonprofit association. We had our first General Assembly back in June, and we have a number of work groups that you can mention or that you can join. You can join our publication group, our training and education working group, communication, HPC and cloud computing. And I really would love to encourage you to actually join our mailing list at minimum, but please consider becoming a member. If the more members we have, the more activities we can do, the more we can keep doing great things like the seminar series that allow us to sort of share bioimage analysis information around the world and really bring this community together. GloBIAS's official purpose is to create a sustainable base and as well as a legal entity for knowledge sharing, networking, and professional development for bioimage analysts, advance the tools and best practices of bioimage analysis, develop and share high-quality educational resources for all skill levels, tackle challenges and seize opportunities, and regularly organize in-person and online events. And our seminar series is here to help everyone stay up to date. We are primarily targeted to intermediate and advanced analysts, but of course everyone is welcome. And we strive to give you practical high-level content that extends beyond basic instruction. And the Bird Institute is helping with infrastructure for this. The seminar will be recorded. Questions may be transferred later to image.sc. Please utilize the question and answer box to send questions to Jean-Yves, and we'll be doing question and answers at the end of his fantastic talk. We will not be having our seminar series next month. Hopefully many of you will be in Kobe, Japan for our first in-person large meeting. But please keep an eye on our website, www.globias.org, for updates. With that, I'm going to stop sharing. And Jean-Yves, whenever you're ready, we're so excited to have you present today.
Let's go. Right. Does this work for you?
Beautiful.
Okay. Beth, Christa, thank you so much for the very nice invitation and the introduction. My name is Jean-Yves Tinevez, and I'm a research engineer working in a core facility of the Institut Pasteur in Paris. And the Institut Pasteur is I would say, a rather large research institute, and our focus is on, fundamental research on human virulent pathogens, basically. So when I mean large, I mean there's about 3,000 people working in 143 research units, and I am part of one of the 32 core facilities dedicated to support the research project of scientists. In the case of pathogens, imaging has a special place. Like for instance, you see here a time-lapse video tour de force made by Laure Leblanc in the lab of Guillaume Duménil. You see a tiny bacteria, Neisseria meningitis, the causative agents of meningitis, growing and in agar pads. So in such a movie, there's really a breadth of information, quantitative information, right? The bacteria are small, but you can see the shape. And it's a live movie, so you can see how the shape evolves. So with that, you can resolve, the cell cycle and the morphological changes in the cell cycle of the bacteria. The movies are long, so you could, for instance, see how the cell cycle lengths evolve over time. And probably with your acute eyes, you notice that they accelerate. The field of view is large, so for instance, you could compare how the cell cycle and the shape are affected comparing the center of a colony versus the outer part and so on. Right, so to extract this quantitative information, you typically need bioimage analysis, right? A set of skills at the intersections of computer science, image analysis, image processing, and imaging. And that's typically not in the curriculum of most of the biologists I work with. And so to address these needs, their needs, and to share expertise, Pasteur has built the Image Analysis Hub. That's the core facility I work with these wonderful people here. So I put a little emoji for Marvin and Marie that just recently departed, and we are very sad. And so our mission is, so we were a research core facility, so we do not have the warrant to do research ourselves. We don't have academic freedom. It's really research support, and our mission is really that, support the research project of others. And we are evaluated by how good we are at that. But to address the need of the campus, we would typically do bioimage analysis, some tool development, and a tiny bit of methods research with these 4 activities, and I will come back to them in a moment. All right, so this is what I will speak about today. The part of the seminar series really focused on research infrastructure for bioimage analysis. I have the difficult task of speaking after an acclaim, and I will do my best to show you how we are organized, how we work with these topics that you see here. I'm not going to speak at all about the tools we develop or the science, but really focus on just the analysis part in the next 40 minutes. Just going to be that. And so hopefully you find this useful for your own endeavors. The first things I'd like to speak about is the fact that we are specialized. The facility was created at the end of 2017, and at the time there were very few dedicated core for image analysis, right? Everybody does image analysis. It's super important, but typically they do that in image analysis cores, in other research labs and so on. There was not that many dedicated course for that. I'd like just to take 5 minutes to discuss with you, the relative benefits and risks of specialization. Why do you want to dedicate a core, a research infrastructure to bioimage analysis? There are many reasons. One of them I will not speak about, is the leveraging, actually, the network part. There's a strong interest in bioimage analysis everywhere, and people tend to recruit engineers that would be isolated in cores. And when you are a dedicated core, you can build the critical mass of engineers that would then favor exchanges of knowledge and know-how. And so this is what we've done in France with my excellent colleagues that you see pictured here. I will not speak about it at all because first we had the chance to make a paper about it. And I was kindly invited by Eurobiomaging and made a recorded presentation about it. I'd like to speak maybe more about the local impact and what does it mean for my users. Well, first, typically, it's when you have a dedicated core not attached to an imaging facility, well, of course, you can analyze the images that come from everywhere in the campus. Now, for instance, here, all of this is project from Dimitri Hershov. That's Helicobacter pylori images. So that's a pathogen. The microscope is going to be in labs in a special BSL-2 plus enclosure. And so it doesn't belong to the core facility. And now, if you have a dedicated core, you can do image analysis for them. Not everything is actually microscopy images. One of the first projects we had were actually satellite images. A researcher asked us to investigate whether there was a relationship between the distance from houses to latrines and kitchens in Bangui in Africa during an epidemic outbreak. And sometimes we do also services for other cores, other core facilities. The microfluidic core once asked us to make the microfluidic core in that case, sorry, an image analysis pipeline used to do quality control on a liquid dispensing robot, right? But this is how you reach to more people on the campus with a dedicated core. Obviously, you don't need to be in a dedicated core to do image analysis, but in our case, it enables us to really focus on a narrower set of concerns. We only talk about, and we only hold the skills that are related to imaging informatics. Now, this involves actually quite a lot of things, right? The mastery of existing tools to develop image analysis pipeline, the mastery of dedicating hardware, high-performance computer clusters, and so on. Programming skills, and so that we will use in turn to create new tools, and actually learning also on image processing. This is applied mathematics and so on. And then also learning to teach, right? Teaching image analysis is super important in our endeavor, and learning to teach is a good idea, right? Again, this would work even if you're not in a dedicated core, but it allows us to really focus on that. And so that's the triptych of skills that I typically use to communicate about what we know in the core. Like if we want to do a good job, we need to know a little bit of, image analysis, what algorithms work for what kind of things, what kind of problems. So it is applied mathematics, computer science as well. Like you want to be able to create tools, to create pipelines, to share with them. So you need to know about programming a little bit. And you need to still know about imaging. And I'm coming to that. Why imaging? Why do you still want to keep imaging in a core facility dedicated to data analysis? Well, when we spun out the imaging core, I was very, let's say, worried that I would cut the ties with my the core facility I came for. The core facility was created a spin-off from the imaging facility in Pasteur, and I thought it was super important to keep the ties with close ties with them, or actually take some skills with them. The reason is that bioimage analysis is tightly coupled to imaging. I think this is especially that's actually the case for every core facility that do some kind of data analysis, right? You always want to have some knowledge about how the data, the image, whatever is acquired. But I think it's especially true for us because every time we have a project, I realize how this is important. For instance, like this is an example of a project on malaria parasites. The research unit that contacted us, they wanted us to create a pipeline that would assess the dynamics of malaria parasites based on static fluorescent images, where the fluorescent parasite they would shed some fluorescent particle along the way. And so we collaborated with our excellent colleagues from the imaging facility, Julien Fernandez, and we created another pipeline based on phase contrast imaging because I knew that you could get a good contrast of these bugs on the microscope. And so now you have something, an image analysis pipeline that is more accurate, but also cheaper in that case, because the malaria parasites that are not fluorescents are cheaper, but also less invasive. And so we want to be able to propose that to our users. And for this, we need to keep in check with our excellent colleagues from imaging. So sometimes, a complicated image analysis project can be made trivial by the right choice of a microscope. So you need to have some knowledge and to keep ties with them. There's also risk if you don't do that. Let's say in frank terms, sometimes the images that you will receive from a user, they might be suboptimal. That happens a lot when the labs own their own microscope and they do not have a skilled microscopist, to care for it. And you need an analyst to be able to recognize that the images could and should be better, actually. Otherwise, you will lose time. But also, as an analyst, I will use the term engineer in this talk. Our mission is really to advise people. And so sometimes, the advice should be, hey, you should not use a confocal. White field is good enough and it's going to accelerate your process and so on. And again, this is important to have skills. And finally, in my humble opinion, working in a core that is self-centered purely on the technical part, it's not so desirable. Like you want to be close to your users, close to the problems. You want to accelerate biology more than image processing. So we are well aware in the core, and so we overcompensate, right? We enjoy a beautiful scientific relationship with the imaging core of the late Spencer Short and Natalie Olner. And this collaboration has been very fruitful scientifically. We built a beautiful perimeter for our activities around light microscopy image, whether it come from light sheets, developmental biology, microbiology, or even non-microscopy images. This perimeter, it's around light microscopy. Personally, that's the one I am at ease with. That's my happy place, if you want. But, as we go, you can guess where we are going, right? Recently, we are having endeavor collaborating with the histopathology of David Hardy, the histopathology core facility of David Hardy to try to offer new services for computational pathology and digital pathology. And even more recently, we've been going towards with my excellent colleague Mohammad Arastani towards EM image analysis, particularly cryo-EM image analysis. And for this, we collaborate with the two facilities here in Pasteur. Right, so this is more or less, the perimeter of our service, how we evolve, how we apply things, and so on. I'd like to spend, the next few minutes in telling how we work and particularly spend some time on our operational model. One of the main difficulty, or let's say challenge I have, is that the Pasteur is a relatively large institute. There's 30 departments, but there's also super varied, right? It comes from neuroscience to mycology, virology, developmental biology, cell biology, chemistry, and I have to tailor my service to all of them, right? And so the core facility model I could use, there was not that many choice that been reasonable. I elected to go for open access, equal access. Basically, it means there's no selection committee. All projects, they are accepted if they are feasible or if the feasibility can be decided. And all of our activities are built, right? So when we work for you, we will charge for our time, which means that the prioritization of the project, the selection of the process, mainly they happen on the user level. They decide what is important for them. And then we have a certain scope. I will come to that in a minute. But the scope of our project, the size of the project we can accept, they are such that we would meant to be collaborating with the research lab in image processing. Now in Pasteur, there are several research labs in image processing, and we are told also that we have to collaborate with them rather than overlap with those skills and so on. And it's been really great. These are the four activities, right? That's these are the services that you can find in the core. Basically, they're very simple. It's about providing autonomous image analysis, walk-in support, consultations, collaborative projects, the development of custom image analysis, and development projects, and so on. For our users, what this means is that typically for the first one, this is simply creating and maintaining workstations with commercial software installed, such as Ivy, Simaris, Huygens, etc. This address actually needs of biologists in well-established pipelines with very good software, with very good user interface and documentations. Consultation, the second one, they address quick questions. Like someone would come and say, hey, how do I do that? What tool would work for this? And so on. And for us, we implement that with OpenDesk, and this is a fantastic tool for streamlined organizations. I will, trying to be lean and efficient. I will come to that in a moment. Collaborative project, I would say that's the main focus of the facility. Like this is the first things people think about. It's when you have a special needs, a tool that doesn't exist, an analysis pipeline that is original, you have to write it, you have to develop it yourself, right? This is what our people expect, our users expect, that we write custom image analysis to address like difficult or non-existing, innovative questions and so on. Finally, per Pasteur rules, like every engineer can dedicate 20% of their time for the development of let's say a tool beyond the scope of a single user project, right? So these four activities, they are split up like this, right? The first three ones, they correspond to articulated requests from our users. Like, someone come, they have a question, and it ends up being any of these three activities, right? Otherwise, we don't do it. And then the fourth one corresponds to unarticulated requests. That is, I feel that the campus needs a special tool, let's say, for microbiology, and we will spend time developing. These four activities, they are sorted by the, let's say, I would say, increasing scientific commitment and increasing scientific responsibility for the engineer. And this help actually addressing, articulating the discussion about acknowledgement, authorships, and so on. Every time, I spend a lot of my time articulating what I do with my scientific direction. And all of these activities, they actually correspond to similar services you can find in other core facilities, even beyond image analysis. For instance, here, the infrastructure part, they support actually economies of scale, right? I will invest me into one expensive software and share it with the whole campus instead of having, each lab buying the license every time, right? The walk-in support and the consultation, they lower the barriers to entry, right? By just speaking with an engineer for analysts 1 hour, they will help you get started yourself. This supports actually elevating the base knowledge of the campus, right? Even if image analysis is not their focus, their main background, with this activity, they will become better at this. Collaborative projects, they're here for removing hard bottlenecks. Research is mediated by innovation, right? Every time, every researcher will have a crazy question that has never been asked before. And of course, likely the tools to address this question doesn't exist at the time. And so we have to build the analysis pipeline to address these specific questions, right? This is what I meant by removing hard bottlenecks. And this is where we share expertise with our users. And then finally, the last one, I will have the chance in a few minutes to develop and elaborate on that. It's typically how you remain scientifically relevant. How do you make sure that the engineers in your team, they progress in skills and so on. Additionally, this is also the future medium of efficiency. How do we answer to user requests faster with the tools we build? Right, I'd like to spend a few minutes on collaborative projects, the third activities we have, right? This is what occupies most of our time, like our everyday time, and typically they're here to address the diversity of bioimage analysis. What surprised me is I do image analysis and we start with light microscopy. And so early on, when I was a young engineer, I thought that I could live with 4 or 5 different pipelines, but absolutely not the case. The creativity of researchers is incredible in imaging, right? Every time there's something new that will completely, let's say, void everything you did before. It might be, analyzing a large microscopy image. This is something that becomes more and more common. Sometimes you have to do image analysis on, let's say, for instance, super resolution images for which there exists very little tools. Here, this is a pipeline where we created a tool that can measure a long filament, the periodicity of a certain doublet. Sometimes people have a crazy geometry in tissues. And here, that's the project of Mylène Lancino. She wanted to measure traction force on cells, except that the cells, they're wrapped around an aorta like this, and the geometry is cylindrical. So, of course, you have to create something like this. Then, there's, everything which is, cryo-EM, cryo-clem image analysis. There's still few new tools that progress quickly. But in the beginning, you have to create everything yourself, right? And so this is what the third activity we have, the collaborative projects exist for. This is for projects for which we have to create something new, an image analysis pipeline. The way we will work is that we will team up with the user and very often with our colleagues from the imaging facility. And an analyst will dedicate some of their time to a specific project. They will work asynchronously on it. They will contribute hours, and in exchange, they will bill the researcher, and the output is that certain deliverables. It might be a tool, it might be results, and it is always a report about what we do. So we've been doing that for quite some time. Roughly, very broadly speaking, the collaborative projects, they fell in 2 types. Either there can be a quantification project. In that case, the user, they would ask you biology questions. And here, for instance, this is where does the bacteria enter in the gut during an infection? And so they expect you to receive a table or figures or like this. Or it can be tool development. In that case, the user, they ask you, please make me a tool that can follow cells in this specific microscope for I built and so on. And here, the user requests, you see, they focus more on the technical aspects. If you remember the triptych I used before, is that typically tool development would require mainly like computer science, the ability to program, develop tools, and quantification, they would require mainly knowledge in image analysis tools, bioimage analysis tools that exist and so on. But of course, everything is blurry in truth. And so if you think about it, this is the main added value we have for our users. We contribute to the research project with skills that they don't have. It's something I say. If you know bioimage analysis, if you know programming and everything, you don't need us at all, right? We're here to support the people that are specialized in biology so much that they do not have these skills. It's been always great. And these collaborative projects, they also have a certain scope nowadays. I try to explain that in this clumsy axis. Let's say that you have a user request, and it would fall on this line that would classify them by the effort, time, or technicity it would require to address them, right? If you have, let's say, something that's low technicity, low time, users, they can always use commercial or open source software. In my humble opinion, compared to a decade ago and so on, the quality is amazing. Like commercial tool, open tools, the documentation, the user friendliness is incredible. And I think we live in kind of the golden age of bioimage analysis tools, all right? So these tools, they would really address everything which is let's say, common, well-established pipelines. At the complete other extreme, if you have something super specific, like that involve a lot of time and effort and creativity, my users, they can go for a collaboration with a research group in image processing. Alors here I put an example of such collaboration with my excellent colleague Jean-Christophe Olivoma from the Institut Pasteur when they developed actually during a PhD a specific technique to address endocytosis. And we, in the core facility, we live in the sweet spot just between, right? So we try to develop something original, highly customized analysis pipeline that would address what I call the mouton à cinq pattes, the super diversity you have and the request for innovative tools and so on. But we try to do that in a timely manner, right? Because we cannot take the 3 years that a PhD in image processing takes, right? And so if you fall out of this sweet spot, maybe the project is not for us, right? Inside the facility, so we are a facility, so we use plenty of management tools. In my case, I took the most, the available actually software for managing that. That is, my facility runs a PPMS from Stratocore instance. And so each engineer is like a microscope, and they write down the time they spend on each project themselves, right? And at the end, that allows us to aggregate the time spent on a project and to do automatic billing so that I don't have to worry about it. Our output, our deliverables, as I say, it's always most of the time code, an image analysis pipeline or a teaching document, something like this, right? And for this, we simply use an internal GitLab hosting service. Marion and Stéphane in the team, they pushed this quite a lot, particularly for R project and Python project. They made a very nice tool that actually compiles the code of a project into a very nice web page that the user can consult. And so you have the reporting of the project and the content of the project that are at the same time. And again, this is super great. Finally, what we found out is the facility was meant to address all the needs of the campus in image analysis. And so we had a lot of requests immediately. We were saturated by requests immediately. And so I found out it was super useful to have a single entry point for all user requests. And for this, we use the open desks. And so every 2 weeks, morning is reserved with all the team. We all go to a dedicated room and we welcome all users. First come, first serves. There's no meeting, no appointments, and so on. And then you will come, you will discuss your project with us, and we will try to address it on site, right? If we can. Actually, so most of the time we can. Again, we enjoy a very good, let's say, tool set in bioimage analysis, and you can get a lot of things done in 1 hour. If we see that goes beyond, we will create a collaborative project or a training request, or to show you how to use the workstation. But that's whatever you want. It's always a single entry point. Now, for us, sorry, for our users, it's great feedback. The 2 weeks interval is perfect for them, and we get a lot of things done in 1 hour. The feedback is generally very positive. For us, what it means, you have a single entry point, so you never lose a request, but also you don't get emails or phone calls anymore. Like, hey, can we set up a meetings and so on? No need for meetings, no need for appointments. Just come next Thursday. All the team will be there. We will discuss all at the time. So it's a true medium of efficiency, and I really like it, right? All right, in the third part of this talk, I'd like to speak a little bit of development projects, and quickly what they mean for a facility, which is, as you have seen, really focused on support. Well, if you remember this slide, I said that we live in this sweet spot where we try to address at the same time creating highly customized pipelines and deliver them in a timely manner. If you think about it, that's impossible, right? I think it's underappreciated, but every time you have to develop, every time you have to program, it takes a huge amount of time. Like developing a tool de novo is long. It's incredibly long. So how do you reconcile that? I think, a partial answer. This is software platforms. I will explain quickly what it is and how they are used to reconcile this problem. And that's our development projects. Right, early on, I said that we have a means to address articulated requests. You have a question and there's an activity for it. But sometimes that's not good enough for researchers. Like what if, for instance, the tool to address a question does not exist? What if you want to be efficient? Like, what if you want to address a class of problems, not just one? And then what is super important in research, I think, sometimes you want to stimulate the researchers of your campus and say, hey, look, I have this new technology. Maybe it could help or could, give you idea for your next research projects and so on. And so for this, there's 4 activities. It's a development projects and so on, in which every engineer can dedicate 20% of their time to creating a scientific tool. And so this is the tools we make. These are, sorry, the tools we make. It looks like the list is very long actually, but many of them are actually already finished. I put, highlighted a few of the active ones. They, again, they correspond to unarticulated project, but they serve really a great purpose. First thing, you will probably notice that there's not truly a specialty here, right? They cover different areas of bioimage analysis. I'll come to that. If anything in Pasteur, we mainly focus on creating tools that are user-friendly, made for end users, right? There must be tools that you can give to a biologist and that they will be able to use it themselves. They must be interactive. This is something really I like. I think it's because I come from microscopy, right? I want to interact with the data, not just with an Excel table. And more and more, particularly with the collaboration with Tobias Pitch and Pavel Tomanchak in the Mammoth and Mastodon projects, we try to address large images. I think that's a recurring problems that we need custom tools for that. And then all of them are software platforms. That means they are not the implementation of one algorithm. There are platforms in which you can incorporate and integrate many algorithms to solve a specific topic in bioimage analysis. I'll come to that in a moment. But, for let's say, at the facility level, they also serve another purpose. These tools, they're always developed in a collaboration with a research group abroad. And I put some examples here, like for instance, sometimes internal collaboration, DeepIC and JDL, for instance, they developed with the lab of Jean-Christophe Olivomin. TrackMate with Kevin Elecheri in USA and Guillaume Jacquemet. Mastodon is with Pavel Tomanchak in Max Planck of Dresden. Multiview Stitcher by Marvin with the lab of Ernst Seltzer and the High Performance Computing Group in Frankfurt. And Clay Esperanto, led by Stefan, it's a collaboration with the unit of Robert Zahab in Dresden and Leipzig, right? So this is fun first and also important because our mission as engineers is to import on campus, technologies and techniques that we do not have on the campus. I think that's a great way to do it, right? If we only collaborate between us, we will never get ahead outside. I think that's a beautiful way to do it. Additionally, the engineers, they get exposed to new techniques. They get trained by their collaborators and get exposed to new techniques. So it's a fantastic way to do that, right? To import new techniques and train your engineers. Now, I need to correct something, right? I said that these development activities, they correspond to unarticulated needs. I have to mitigate that a little bit. If I take, some of the graph that you see before, for instance, localized projector from Sebastian and Selige from Céline, they actually span from 4 to 6 different projects that we have in the developmental biology and here in Institute, the Institut Pasteur. So they are not so articulated. The same come from the multiview stitcher of Marvin. Actually, it became absolutely critical for a cryo-clamp pipeline that was published recently. And now it's super important for spatialomics and metabolomics pipelines and so on. And finally, there's a new version of TrackMate coming and a new track analysis library made by Marie and then Laura, respectively. They correspond to actually new needs from the microbiology department, like probably 8, 10 projects come with people that want to image and track bacteria over time. I will not speak about it at all. If you ever come to the C-Bias conference in the Creek, London, I will have a chance to present it, hopefully, right? So they are not so unarticulated. There's always something that can be tied up to the campus directly. And finally, they are super important for the analyst and for the engineer. If you think about it, like in such a facility, you have no equipment, you have no microscope, I have no big machines. The only thing I have are talented engineers. All the quality of the services I deliver are made via the skills of these engineers. So you absolutely want to have a means to, let's say, own these skills. And a development project is a really great way to do that. Now here I put a little of a extreme example with Céline Trébois. Sorry, I forgot to put a nice picture. Céline does not directly work in the facilities. That's a collaboration with units Raphaël Tourné. At some point, they discovered that they needed a way to extract tissue manifold from 3D images where the tissues were really damaged. Here, that's an example from the COVID, or from cochlea, where actually the tissue manifold are stacked over each other. So Céline took this development project, she taught herself to program, she taught herself algorithms, and she created Zelige, the tool, and that was published a couple of years ago here. And of course, she used that as a medium to progress, like to learn new skills, programming, image analysis, algorithms, etc. But this materialized quite extreme because she used this to get promoted from technician. Now she's a research engineer. So that's super important, right? The development projects, there's a mean to build the skills of your analysts. And without that, you take the risk of stagnating, actually. Right. And then finally, the development projects, they serve another purpose, right? So I'm a very lucky person. When I run my yearly user survey, generally the results are very good. People are very happy with the quality and everything, right? And this is an excerpt from 2019 user survey. And this question focused on the delays. How much time does it take to start a user project? And we have 1% that is very unhappy. I put it literally, it's in French, means I wait for too long, science doesn't wait. And so what our users value when they have everything, they actually value having the pipeline quickly. Now, that was surprising for me, because programming takes so much time, but no, they want it quickly. I think the reason is that the image analysis is critical early and late in the project, right? For instance, you will need image analysis to know whether a subtle phenotype is here or not, and this will orient your project. And image analysis, is, as I said before, it's not in a vacuum, it's in a closed loop with the imaging. So the image analysis results, they will guide sample preparation imaging. So you want to know if the image analysis can give you the results you want, or if you have to go back to the wet lab and create samples. And so typically image analysis is done early in the project. And typically we will hear something like this: I cannot go on with your pipeline, please deliver it quickly. Or late in the project, and that's the more classical stuff. I'm sure we all know. I'm defending next week, please, quick. Bon, so they value time. How do we address that? Well, it's very difficult. But here, the software platforms, they are really useful. This is a very old project, one of the first we got. This is an assay in immunology. And the researchers, they have this beautiful, let's say, imaging setup in which they mix B cells that you see here, showing with the finger is not very useful, that you see here in red, with T cells stained in green. And so they will form synapses or even synapses. And when this happens, the T cells will activate and sense a calcium flash. And so what the PhD, the student asked me, say, please create a pipeline that can automatically, from these movies, detect the formation of a contact, track them over time, measure the size over time, and then correlate it with, yes or no, I have a calcium pulse. So this is fairly specific, right? And that would be typically a very long project. The way I addressed it was to simply create a small TrackMate module. And something that would just do one thing, that is detecting the contacts. And I did it very simply. This is what you see, and something that simply measure the overlap between the two channels and create a signal. And then, I reuse what was already existing in TrackMate, the tracking modules, let's say, the user interface, the visualization of results, the input-output to save results, etc. And so programming this, creating, let's say, a prototype of this pipeline was very quick, and I could deliver it early on at the beginning of the PhD. Now, in that case, I always remember the story because delivering it early was important, because they run the pipeline and then they found out that the project doesn't work. They are not looking at the right genes, and they completely changed the project and did something else. I always tell myself, all right, what happened? What would have happened if I took 3 years to produce the tools, right? If I produced the tools at the end of the PhD, that would have been harsh, right? And so the importance of these scientific platforms, something that you can quickly extend, something that you can write innovative pipeline is, I think it's super important. Here, I put an example where the scientific platform is TrackMate. It turns out that I'm one of the authors of this, but it doesn't matter. As an analyst, the only thing that matters is that you have a couple, maybe let's say more than a couple of scientific platforms that you master very well and that you can use very quickly to prototype things and so on. And that's actually key to address the timely delivery of your users. All right, so I will finish quickly by giving you some numbers that we had. Like you were asking, okay, here it's written 7 years. I feel like it's been more like 8 or even 16. What have we learned, right? What can I share with you that would be useful, for your endeavor? If you remember, I told you that we have for the facility management, we're using a software in which you can write log the time we spend on project, and over the course of the 8 years, it's generating a large amount of data on which we can reason, right, and so on. And my excellent colleague Stéphane Rigaud created, a set of tools that can extract plenty all information you have. I have like graphs like this by the dozens and so on. They are maybe not so useful to you. I think more useful to me, to understand what's going on in the facility if something's wrong. Here, that's an example of such a plot where that's the timeline of all the projects, right? And so one horizontal line is a project from when the project is created to when it's closed. And what you can learn from this is that typically projects are long, the years are here. And having a project that lasts more than 1 year is kind of surprising for us. Most of the projects they're created early on. For instance, when a PhD joins the lab says, I want to have a project with you. And then they will be closed very late, when the projects here published, which takes very long. Here we could use it to analyze the rate of project opening. Now that we're going into statistics, I'm going to tell you that it's biased because the project opening in my facility, it's actually hindered by the availability of engineers. This means, when an engineer has no time, they don't create new projects. So you wouldn't see it here. And so this dent here, this inflection in the curve, this is caused by, we created plenty of projects and now we have no capacity anymore. And so it started again because in the facility, Marvin, Mari, and most recently Mohammed joined. And so we had a burst and so on, but we will pay the price soon and so on. And then what I'm looking forward to is that now, we are opening new services. I can say, voicing to people, hey, if you have histopathology, you can give us a project. Hey, if you have EM, you can give us a project. So there's going to be probably more. It also helps us to make sense of our capacity and the demand to the facility here. So here, what I plotted, this is all of our projects sorted by end date. And in y-axis, this is the project duration, from start to closing date and so on. Bon, you see that there's a clear trend. You can see the effect of the COVID here. But roughly speaking, now it's more and more common to have projects that are, longer than 1 year. I don't know about you, but I have the feeling that since 4 or 5 years, the ambition of our users are ramped up. They ask us for things that are harder and harder, longer and longer. You can see that as well, with the project effort. This is how much time we bill to our users. You see that, most projects, they are like around 200, 500 hours of work. Well, this is significant, right? Because we tend to underbill our users. And so the work behind this dot is actually quite intense. And then for me and for you, I can use that to actually make estimates about my core facility, right? And to talk to my scientific direction and to my users to explain them. And for instance, we can have this exercise of estimating the capacity of a core facility. How many projects can you accept per year and so on. So here, for instance, I have, just move this out of the way. I have a certain project durations, mean and standard deviation. So you must note that my project durations, it's not a Gaussian distribution, it's actually elbow curve. There's a lot of small projects, either because they close earlier, because they're easy. So like, let's say that it's not multimodal. But I also have an estimate of the project effort, right? And if I take the median, it's about 200 hours. From that, you can estimate the engineer nominal capacity. How much time as an engineer per year? Now in France, we have 226 working days, and I said that each engineer would dedicate 50% of their time to the collaborative project activity. In a day, there's 6 hours that are billable per day when there's no interruption. Let's say that it's a conservative average, which gives you 678 project hours per year. So as a consequence, if you have one engineer, they can work on about 4 projects per year. And then I was very surprised to find out that in the image analysis hub, it's much more than that. It's more like 5, 10, and recently we are edging on the 10 and beyond. I think that my facility is a bit too small for the need I have, and so we've been sacrificing this, right? It's not 50%, it's much more than that, and we've been sacrificing the other activity just to fulfill our users' request. Again, nonetheless, the statistics probably not right. I would have, to compensate by the fact that the project is not one year exactly, that my distribution is an elbow. Probably statisticians know that better than me, but this gives you an estimate, right? You cannot hope to have an engineer do 50 projects per year. That's unrealistic. On the other side, you can also estimate the demands. For instance, in Pasteur, I have 143 units, and every year we support 50 different of them per year. It's not that there's 1/3 that uses the 50 units, they rotate, right? And so in a couple of years, I hope to have sampled all of them. And from consultations, these 50 units per year, they will make about 100, a bit more, 120 requests for consultation per year. I note that the capacity of an engineer that dedicate 10% to this consultation activity can make 25 consultations per year. So up, you need 4 engineers to address that. On a project, you can do the same. If I take the average from 2018 to 2024, I've noticed that we have on average 20 project requests per year. And so to satisfy them all, I would need 4.5 engineers. And again, there's a big footnote here is that the project creation requests, again, as I said, are mitigated by engineer availability. But again, this is something you can use in your own core facility to articulate, size your facility, and even estimate the cost, right? How much such a facility would cost, because you have to pay for the salaries of this person, and knowing that they would be spread over 50 units in a year. And at the same time, with this length, if you have an hourly fees, you can estimate how much a project will cost to a research unit on average. This is kind of useful. So that's my last slide, actually. This is my non-quantitative insight on my job. First thing, like projects are super iterative. Everything will change from one week to another. Come say, hey, I have tested a new probe. Hey, the microscope doesn't work. I change the microscope and so on. So you need to be ready for that. It's super hard to predict the end date. And all the curves and the statistics you've seen, the main reason to do that was that we are trying to guess in advance how much time it will take. Super important and super happy. People involve us early sometime, and that's very pleasant. They come to us and say, in 1 month, I would like to make this experiment, that and this. What should I do? How many samples into that? So they involve us in experimental design. That's great. And that's an opportunity, to create links with our colleagues from the other core facilities. Reporting, I'm very keen on having people write reports about the project. It's great because this is when you find big mistakes and important things. It takes a lot of time. And I try to convince people that this time should be built too, but that's difficult because it's even more immaterial. About charging, for me, charging is absolutely crucial. Like, since I do not prioritize projects, there's no selection committee. You need some way to share the risk with the user, right? And the fact that you would charge their time for them to say, okay, I will give you a project request, but for something that is really important for me. I don't know about you, I found out it's super hard to get acknowledgement because consultations and the usage of workstation is so painless that it doesn't write in the memory of people, but never mind. I'm very lucky, however, because in Pasteur there's a very good culture of collaborations, and so we have no problem for collaborative projects. Again, what I say, projects are done quicker when you base them on scientific platforms. The sad part, people write you only when things do not work. When something, I wish sometimes like people say, hey, Jean-Yves, just write you to let you know that your analysis pipeline works great. There's no need to answer this email. There's no need to set up a meeting. It just works. That doesn't happen, but that's life. And well, that's a minor point. What we call the projects can be part of a larger scientific projects, and so we must be ready for that. Image analysis is very transverse. It's not the privilege of cell biology, of developmental biology. There are very creative people in virology, mycology that will do crazy, awesome stuff with imaging. And so we should be ready for that as well. And finally, that's my last slide. Never mind the statistics, right? A core facility, the way it's organized, it's just a means, right? It's just an organization. In the end, the ultimate outcome is always science. It's always helping others, to create publications and to achieve beautiful new knowledge and science. So we should never forget that. All the rules, all the statistics should be broken and must be broken if it's for the achievement of the greater good in science. And that's what I truly believe in. Thank you very much for your attention. Right, this is my team. That's an old pictures already. Marvin left, Marie left. And they are really awesome, hardworking people. They deserve more than a thing. Like they deserve like a tribute, and that's fantastic to work with them. Like, my job is really cool. I also like to thank you, Nathalie Olner and the late Spencer Short. They really helped me creating the facility. Now Nathalie works with Florian Muller in the facility. It's a pleasure to have them as colleagues. Jean-Christophe Oliveaumarin was the director of the facilities. He gave the impulse to create the facilities that I've talked to you about today. I'm really grateful for them. This is my awesome steering committee and the awesome people that found the facility. I'm sorry to be so long. Thank you very much for your attention.
That was fantastic. Thank you so much.
You're very kind.
We have a number of questions in the chat. I think we'll do as many as we have time for, and then if uh, hopefully be able to sort of do the rest of them on image.sc if that works for you.
All right, let's go.
Um, uh, so, question from Augustine Corbat. You discussed the close ties between imaging and analysis. How does this work in practice? Do you keep the bioimaging facility in the loop when discussing projects? Do you discuss experimental design? And when do you get looped in? At the planning phase, the pilot phase, the late phase?
Everything happens. I couldn't find a good way to streamline that. We are lucky because we are in the same space, right? So this is my future office, and on the floor there's the light microscopy facility. Under the floor there's the electron microscopy floor. We know each other, and because we are in the technology department, we get a chance to exchange a lot. And so it's really proximity that works, and it's ad hoc, typically, right? Also, things are very flexible. Like, for instance, if in one of my open desk, someone comes with a question and we see the image and say, hey, do you mind if I get Audrey or Julien and so on? Can we discuss imaging and so on? That works. That's our job. So we know that we need to be available anytime, roughly speaking, for this kind of question.
Yeah. We had a couple of questions from Tisha and Guillaume. Uh, since it's hard to estimate how long a project will take in terms of hours, how does that work in terms of billing? You say to somebody, it might be this many hours, it might be that many hours. How do you actually manage this uncertainty when you're billing people?
I bill at fixed interval, spring, summer, autumn, winter. I simply, take how many hours have been billed on the project, and then I send the bill. And so all the research units in Pasteur, they receive a global bill for all the usage of all facilities and so on. And I'm just one of them. And, this is how it works.
But do you
Generally,
need
I try to give, rough estimates. at the
Mhm.
beginning of a project based on the numbers you've seen and my intuition. And then I say, any ambitious project, any something significant is going to take you 100 to 500 hours, and that gives you a ratio. And given the fact that it's going to last 1 to 2 years, you're going to be built that much per year, roughly that.
Mhm. And generally speaking, people are okay with that.
No, actually, everybody wish that everything would be for free, right? The C part, but again, I think I'm lucky because beyond that, it always works.
Mhm.
Like the exceptions, they are very, very rare. Where people complain or say, I will not pay this bill, it's extremely rare.
Um, a question from Kenneth Ho. Do you have problems getting groups to acknowledge you or give you co-authorships? He said that's rare for billing, but how about there?
No, I'm lucky. I do not have problems. I think, I've been discussing with a lot of my colleagues. I have the feeling, Bess, you will have to confirm that that situation is better now than 10 years ago at the time of NEUBIAS.
Mhm.
But I'm lucky. In Pasteur, there's a strong culture of core facilities. People are used to have, to use, and to acknowledge core facilities. And our scientific direction, they made a lot of effort in actually making your charter, good practices, and so on.
Mhm.
And no, there's very, very, again, very rare when you have problems. I have only issues with acknowledgements, like, when people use your workstations or use your advice and so on, but, honestly, that's okay. I think that you think that's difficult, right? Because this is immaterial. Like, it's not an access to a microscope, it's not a consumable. As soon as it's immaterial, we're used to MP3, everything that can be downloaded should be for free. But and it should be also difficult because this is not their main focus, right? It's hard to estimate how hard it is to develop a pipeline if you're in biology and need my help. But yeah, I think I'm lucky because of the culture, local culture. The dean, the directors, and so on, they have created this culture and they say, we invest in core facilities, you need to invest in them via this one.
Great. We had a couple of questions around the role of multiple image analysts. Are people generally working on projects together? Does everyone sort of work on, are projects usually assigned to one person? How do they interact with each other around sharing experience and things like that?
So in the image analysis facility, every analyst has the ownership of a project. So there's one analyst, one project, and actually one face to the user. After that, they're free to do everything they want, collaborate with others, but that doesn't happen so much, right? There's one.
Mhm.
It's not optimal, to be honest. Not everybody is happy about it. And if you go this way, you need to compensate by other ways. You need to keep the whole team informed of what everyone's doing. And so for this, we need to set up lab meetings, retreats, whether this happens, or informal talks. Being all at the same place helps. But having one analyst per project, it's important to be efficient and fast. And unfortunately for me, fast is for my users, being fast is important.
Mhm. Um, that's a great segue to another question from Guillaume. You mentioned that you accept all requests without sort of selection. How do you manage to have a reasonable workload? Do you plan projects over longer timescales to even out the burden?
I don't manage. It's we are ah, that's my main challenge, right? I think the workload, I even have a graph for that. The workload we have is too high for the capacity we have. And so I would say that I think we are overworked. And the typical rule I say is that for every engineer, you cannot have more than 3 projects active. And you're forbidden to accept any new one if that's the situation. And so then, if you ever come to Pasteur, on my window, there's this bunch of post-its, and that's the projects that are waiting for an analyst to be free and so on. And I would say that's it.
Yeah. Um, what do people who've worked at the core facility tend to do in terms of jobs?
I wish they stayed in core facility. Actually, you remember the triptych I used when I said the three skills, like programming, applied mathematics, computer science, and imaging? These are three jobs that are in high demand. And so the people that left the facilities, there was always, for, I would say, big endeavor. Dimitri went into a medtech in Russia and created image analysis pipelines here. Marie joined a big company and she has a team where she does a very similar job. Marvin joined the facility of Martin Horn, for instance, and in Switzerland. What can I say? Right now, there's a lot of opportunities, right? Either in the academia, because new core facilities are emerging, or in the industry with very, very little downtime, I would say.
I do just want to recognize that we're at the hour. Anybody who needs to hop off, thank you so much for the gift of your time. We have three questions left. If Jean-Yves, if you have a few minutes, and if it's okay, we'll keep going through the rest of them.
I,
But
I apologize for speaking for so long. I'm perfectly available.
yeah, great. Question from Marco. Thank you for the great talk. How much of the project and code you have in your internal GitLab instance eventually becomes open and public to everyone? Do you strive for all of the code to be public or only for major software that comes with publications?
That's a very good question, under which there are many questions, such as how many of our projects end up successfully? Right? Because you can get like, I think we have more than 200, much more than 200 projects since 8 years. And I think we have about 50 publications with our users. And so, of course, not every project has a positive outcome. Among these 50 projects, every time we can, we open and make the GitLab instance public. And you can find it actually. There's a IH, Image Analysis GitLab, in which you find the analysis pipeline for the specific publications that were made. And then when there's actually a specific tool, for instance, TrackMate was created, for several projects. Everything we do join the code base, right? So you can find it on GitHub or something like this. And we as much as we can, we try to publish everything, which is also important for visibility. And I think that as soon as a paper is out, there's nothing private.
That makes a ton of sense. Um, I'm interested to know how you analyze bioimages from another core, from another institute. How do you access images that might be terabytes in size across the country?
I don't have a good answer for that. The projects we have with FBS that are typically external, so far they weren't that large, not terabyte, gigabytes and so on, for which the transfer via internet exchanging files were fine. I think, however, the future lies in centralized image database with this new and exciting OME and GFF file format, where the data could be there and then streamed to users and analysts alike. But we haven't had a chance to play a lot with that. But that's just me, right? If you interview my colleagues, you will get something different.
Yeah, that is hopefully the future. All right. Last question is from Mike Nelson, is one that I like a lot. How do code resources like image.sc posts or chatbots impact your daily life now? Do you see a lot of messy code generated that takes extra time to troubleshoot, or are they overall time savers for the users and for the analysts?
Huh, alors, I have one case where actually a postdoc came to me and say, I made a code with vibe coding using ChatGPT to measure mean square displacement analysis curves. You see what it is? You track the motility of cells
Hmm.
and you use MSD curve to determine whether they're static or actively traveling. And with the code he wrote, he had the exact opposite answer because of sorting things like this and so on. And me, for the facility, the policy is the following, right? So we will write the code. We will not review your code.
Mhm.
So if you can write the code yourself, you take the responsibility yourself. But what we receive is typically a scientific question, like please build the image analysis pipeline for me. And so this is what we do. So we don't review someone else's code, to be honest. After that, do we ourselves use ChatGPT and Copilot? I would say yes, a lot.
Mhm.
For me, I'm an old person. I started coding with MATLAB and Java. And now when I do Python and teach myself Python libraries, for instance, I find it super useful.
Great. Well, thank you everybody for your attendance and for all of the great questions. It's been lovely to have you, Jean-Yves. This has been great. We look forward to being able to share this with folks on YouTube. And thank you so much, everybody.
Thank you all, really.
We
Thank
will,
you.
yeah, we look forward to seeing everybody back in November.